Hi guys, welcome to Chapter 15, the chromosomal basis of inheritance. In Chapter 15 we will talk about such phenomena such as sex-linked traits and how traits are inherited based on independent assortment and the laws of segregation. So to begin with, let's look at an overview locating genes along chromosomes. So Mendel's heredity factors were genes, though this wasn't known at the time. The location of a particular gene can be seen by tagging isolated chromosomes with a fluorescent dye that highlights the gene. In 15.1, we will discuss Mendelian inheritance has its own physical basis in the behavior of chromosomes. The chromosome theory of inheritance states Mendelian genes have specific loci or positions on chromosomes, and chromosomes undergo segregation and independent assortment. The behavior of chromosomes during meiosis was said to account for Mendel's laws of segregation and independent assortment. Okay, this figure here is showing how we can correlate the results of one of Mendel's dihybrid crosses with the behavior of chromosomes during meiosis. The arrangement of chromosomes at metaphase 1 of meiosis and their movement during anaphase 1 account for the segregation and independent assortment of alleles for seed color and shape. Each cell that undergoes meiosis in an F1 plant produces two kinds of gametes. If we count the results for all cells, however, each F1 plant produces equal numbers of all four kinds of gametes because the alternative chromosome arrangements at metaphase 1 are equally likely. Alright, so let's take a closer look. Alright, the way your chromosomes, alright, if you have heterozygote parent cells going in at the end of meiosis 1 going into meiosis 2, your chromosomes will line up at the center of your cell. From here we can see how the two alleles for each gene separate during gamete formation. Okay, showing the gamete separate. Alright, what the law of independent assortment is showing us is that alleles of non-homologous chromosomes, these are non-homologous chromosomes, alright, and they assort independently during gamete formation. Meaning, just because the seed is yellow does not have to be round, just because it's wrinkled does not have to be green. They can assort independently of each other. They don't have to travel together. Okay? So, how they line up at metaphase and how the chromosomes sort during anaphase and into telophase to produce your new gametes all depends on how those chromosomes segregate from each other. Okay? Does this make sense? Notice to the left you have your chromosome pairs, your homologous pairs lined up in the same order, red to blue. And so when they segregate, when they separate from each other, one daughter cell will end up with all red, in this case red chromosomes, one will end up with all blue. And so when everything is divided out to reach your gametes, it is possible to end up with only, in this case might be dominant traits, okay, in the chromosomal structure of the gametes, and one with only recessives. However, your other cell over here, your homologous pairs are not lined up in the same order. It's not red to blue in both cases. It's blue to red, then red to blue. So when your segregation occurs in your cell, your chromosomes, you have a different combination of gametes. They end up with a heterozygote amount of different chromosomes. They end up with dominant and recessive alleles, for example, with this trait specifically. Okay? If that doesn't make sense, let me know. We'll talk more about that. Alright, Morgan's experimental evidence of choice of experimental organism. Morgan's experiments with the fruit flies provided convincing evidence that chromosomes are the location of Mendel's heritable factors. Several characteristics make fruit flies a convenient organism for genetic studies. They breed at a high rate. A generation can be bred every 2 weeks, and they have only 4 pair of chromosomes. Alright, in my genetics studies as an undergrad, worked with fruit flies myself, and they reach sexual maturity within 8 hours, so they are very, very easy to use for such study to see how traits are inherited, because you can rapidly create your parental F1 and F2 generations to see how traits are inherited. With the fruit flies that I worked with, where I crossed wild type fruit flies, which I'll show you in a moment, one of the things, which had red eyes and normal round wings, and my mutant flies had white eyes and I called them curly wings, but they had a lobe to them, like a lobed wing. Okay? And so I studied how those traits are inherited. In my case, it was simple Mendelian inheritance, because everything occurred in a 3-to-1 ratio. So nothing was sex-linked. There was no polygenic inheritance. It was straightforward for me. But other people had differences such as antenna shape, body color. Those things could be sex-linked or related or inherited in another pattern than simple Mendelian inheritance. So you had to conduct your study through the F2 generation with multiple F2s to be able to see how many offspring ended up with which traits. Our F1 generation had to consist of 200 flies. Our F2 had to consist of 400 fruit flies. It was tedious, to say the least. Okay? They're showing the phenotypes of fruit fly eye options. Your wild type are red. That is your standard that you would find in nature. Your mutant that has had a mutated allele has white eyes in this case, and this was also the trait, one of the traits that I studied as an undergrad. Okay. Alright, so they correlated behavior of a gene's alleles with the behavior of a chromosome pair. So in one of their experiments, Morgan mated male flies with white eyes, the mutant, and female flies with red eyes, the wild type fly. The F1 generation all had red eyes, and the F2 generation showed the 3-to-1 red to white eye ratio. Before we talk about that next part, that alone says simple, complete dominance. Alright, complete dominance for the red color. However, they found that only the males had white eyes. So what that clued them into was that the white-eyed mutant allele had to be an X-linked gene. It had to be on the X chromosome. So that would be a sex-linked trait, okay? So Morgan's findings supported that chromosomal theory of inheritance. Okay, this showing that X-linked eye color, if he bred a red-eyed female to a white-eyed mutant male, all of their F1s have red eyes. They bred 2 F1s to end up with their F2 generation. They end up with that 3-to-1 ratio, which shows simple dominance, alright, and recessiveness. And we can find that the red eye trait is dominant to white eyes. However, it's curious that the white eye individuals were only males. So, what they concluded is the trait for white eyes, which was first introduced by the father, is carried on the X chromosome. So when this lowercase w by itself here was passed to the F1 generation, one of the female F1s became a carrier of that trait. Now, it did not exhibit because the dominant trait, the red-eye trait, masked that white-eyed mutant trait, okay, masked that phenotype. So when the F1s were allowed to mate, the F2 generation ended up with a female carrying the white-eyed trait. However, the red-eye trait is dominant, so it was what was phenotypically expressed, and one male ended up with the white-eye trait, which, that being their only X, there was nothing to mask it. It was expressed, so you end up with one white-eyed male. Alright? Pretty straightforward. You just have to think it through. Let me know if you need help on that. Okay? Alright, so in 15.2, they talk about sex-linked traits, carrying that conversation that they started with the flies and their eye color. Alright, they carry that conversation on with talking about how sex-linked genes exhibit unique patterns of inheritance, this chromosomal basis of sex. Alright, in humans and other mammals, there are two varieties of sex chromosomes, a larger X and a smaller Y chromosome. Alright, at the ends of the Y chromosome, they have regions that are homologous with the X chromosome. The SRY gene on the Y chromosome codes for the development of testes. Okay? This is where your determinations of sex for different species fall. With humans, we have the XY system, so your female is XX, your male is XY. Alright? Your grasshoppers and other such insects have an X0 system, so meaning female would have two X's and male would only carry one. Okay? This one, the example it gives is in some birds and some types of fish. The sex chromosome present in the egg, the sex chromosome that's present in the egg, not the sperm, determines the sex of the offspring. So their sex chromosomes are designated Z and W. Females are ZW, males are ZZ. Okay? Alright, and then the last one, the haplodiploid system. There are no sex chromosomes in most species of bees and ants. So females develop from fertilized eggs and are thus diploid. Males develop from unfertilized eggs and are haploid. They have no fathers. Okay? Does that make sense? Males don't have a father. They are just a haploid organism. They develop from a haploid cell. Slightly confusing. Very interesting. Okay? Alright, the inheritance of sex-linked genes. The sex chromosomes have genes for many characters unrelated to sex. A gene located on either sex chromosome is called a sex-linked gene. In humans, sex-linked usually refers to a gene on the larger X chromosome. So things like hemophilia, color blindness, those are both located on that X chromosome. Okay? Sex-linked genes follow specific patterns of inheritance. For a recessive sex-linked trait to be expressed, the female would need two copies of the allele, because as long as there's a dominant copy, it will be what's expressed. And for a male, they only need one copy of the allele because they only have one X, so there's nothing else to phenotypically express. So that will be what is shown. Alright? Sex-linked recessive disorders are much more common in males than females, such as color blindness. Okay? And that's because with a female, the sex-linked disorder would have to be carried by both mother and father. Would have to have at least a carrier mother and a father that is expressing that sex-linked trait for the female to be able to get it, to carry that trait, to have that trait as well. Because if the mother is not at least heterozygous for the trait, then they'll inherit at least one normal X and thus the other will just be masculine, then they'd just be considered a carrier. Okay? So, in this example, uppercase N is our normal dominant, lowercase n is disorder, is recessive. Okay? So, when you look at these three examples, the female is normal. She does not carry the disorder on either of her X's. The male carries the recessive sex-linked disorder, on his X chromosome. So when those two parents have children, we'll use people for example here, when those parents have children, they get a normal X from mom because that's all she has to give, and they'll get, in female children, they'll get that recessive disorder trait from dad, because that's his only X to give, and then the males will get the Y from dad, and they will not be affected at all. The females will be now what you would call a carrier, because they do carry that trait on one of their X chromosomes. Okay? In example B over here, mom is a carrier. She has one normal and one affected X. Dad is absolutely normal. He does not have an affected X. So, depending on which X they get from mom, kids could be completely normal, getting the normal X from mom and dad's normal X, so normal daughter. Could have a normal son, get the normal X from mom, get dad's Y, no issues there. Could have a daughter that is a carrier, get the affected X from mom, dad's regular X, or you could have a son that is affected, get mom's affected X chromosome and dad's Y, so that trait would express. Okay? And in example 3, you have a carrier mom and an affected dad, so the way the kids would pan out, one daughter would get mom's normal X, dad's affected N, be a carrier as well. One son would get mom's normal X and dad's Y, would not be affected at all by the trait. One daughter would get mom's affected X, dad's affected X, and exhibit this sex-linked disorder. And then one son would have a chance of getting the affected X from mom and dad's Y, so they would also be affected by this disorder. Alright? Does that make sense? This goes into a lot of when we talk about pedigrees. Being able to trace disorders through a family tree, you can do this with a pedigree. If you have questions on those when we get together again, we will talk more about this. If you want to hash this out some more, have questions, this is discussed on page 291. Okay? The trait that they used here was color blindness. I'm sorry I never mentioned what it was, but with any recessive sex-linked trait, this is how it would function. Okay? So, to continue, alright, human sex-linked disorders. Some disorders caused by recessive alleles on the X chromosome in humans include color blindness, forgive me if I pronounce this wrong, Duchenne muscular dystrophy, which affects 1 out of every 3,500 males born in the United States. It's characterized by progressive weakening of the muscles and loss of coordination. Affected individuals rarely live past their early 20s. Researchers have traced the disorder to the absence of a key muscle protein called dystrophin, and have mapped the gene for this protein to a specific locus on the X chromosome. Okay? And then hemophilia, which we've discussed a little before, sex-linked recessive disorder defined by the absence of one or more proteins required for blood clotting. A person with hemophilia gets injured, bleeding would be prolonged because a firm clot is slow to form. Small cuts in the skin are usually not a problem, but bleeding in the muscles or joints can be painful and lead to serious damage. Today, people with hemophilia are treated as needed with intravenous injections of that missing protein. Back before that missing protein was available, it used to be a lot more serious of a disorder, and it was very prevalent in the royal family due to inbreeding back in the Victorian ages. Okay, so let's keep going. Alright, X inactivation in female mammals in mammalian females, one of the two X chromosomes in each cell is randomly inactivated during embryonic development. The inactive X condenses into a bar body. Okay. Alright. A bar body lies along the inside of the nuclear envelope. Most of the genes of the X chromosome that forms the bar body are not expressed. Alright. In the ovaries, bar body chromosomes are reactivated in the cells that give rise to eggs, so every female gamete has an active X. Okay? If a female is heterozygous for a particular gene located on the X chromosome, she will be a mosaic for that character. Alright. Let's see. Is there anything I want to add about that? Alright, that's it for now. We'll come back to that. Alright, X inactivation is random in female mammal cells. Alright, so you have an allele. This is, use a cat for example. At the allele for orange fur, alright, on your X chromosome, and you have an allele for black fur on your X chromosome. So when cell division and chromosome inactivation take place, you have two cell populations in the adult cat. An active X for black fur, and then inactive X has orange, and then the opposite, your active X has orange fur, inactive has black. And that's when they're a mosaic for the character because they are both expressed here. Okay. This is that tortoiseshell gene. It's on that X chromosome, and tortoiseshell phenotype requires the presence of two different alleles, one for orange fur and one for black. Normally, only females can have both alleles because only they have two X chromosomes. So if a female is heterozygous for the tortoiseshell gene, she is tortoiseshell. Alright, if female, it'll have orange patches formed by population of cells with the X chromosome with orange allele is active. Black patches will have an active black allele. Calico cats also have a white area, which is determined by yet another gene. That's sufficient for now. Just know that X inactivation, it is random, but so for this cat being heterozygous and having this these bar bodies, those inactive X's, within the cells that determine coat color, they end up with a combination. They end up being a mosaic for those traits, okay? We'll keep going. Oh, no, we're not. We're done. We're going to stop right here at 15.3. We'll pick up here next. If you have any questions, please let me know. For the sake of time, I'm going to lump your question into just your checkpoint into just one sheet. So just make sure you watch all of your videos and then answer your questions. Let me know of anything that you do not understand. Thanks, guys. Bye.